and I will briefly introduce you. So is there anything that I will see here, Marcio, as a...
Tá tudo certo, Gu. That's all fine.
Can I start now? All right, so welcome to our last session of this graduate course on molecular control of the different forms of cell death. This is my great pleasure to introduce a speaker that needs no introduction whatsoever, as Doug is one of the most cited and honored scientists in the field of cell death, immunology, molecular biology, tumor biology, so on and so forth. He was indeed formally recognized by the Web of Science in 2020 as one of the most cited scientists of all times. So guys, enjoy this last conference. But much more than that, Doug is a real great friend. He was my mentor, and he's a very kind and generous person that really inspires everyone around. So it's a really pity that we don't get together more often, Doug, but we will soon, make arrangements to do that. Anyway, just to get a little bit on some formalities, Professor Doug Green is the Peter Doherty Endowed Chair of Immunology, the chair of the Department of Immunology, and the co-leader of the Cancer Biology Program at St. Jude's Children's Research Hospital in Memphis. Doug is also a distinguished fellow of the American Association of Immunologists, member of the U.S. National Academy of Science, and a foreign fellow of the Royal Society of Canada. Doug is now or was at the editorial board of the most important journals in the field of biomedical science. So yeah, students, prepare for this awesome experience and receive Doug's inspiration and learn about the future of the cell death field. Okay, Doug, thanks a million again for being such a great friend and being a long life collaborator.
Thanks, Doug.
And Karina, thank you so much. And
thank you.
Really, it's so much fun to participate in this, and hopefully we'll do it next time in person. All of the data that I'm going to show you today is unpublished, so please, and I'll say this a few times, please don't take a picture of it and send it to your friends and put it up on the web, because my people work so hard on this stuff, and we don't want to get surprised by some little publication that comes out in 2 weeks. So let's go cell death into the future, if it'll actually progress. Come on. Here we go. Cell death. My relationship with death remains the same. I am strongly against it. So we're going to try to predict the future. And you may have seen in movies like the movie Big with Tom Hanks, and sometimes at amusement parks, there's this thing, a machine called Zoltar, and you put in $1 and it gives you your fortune. So that's what I did. I asked Zoltar a bunch of times, more than 5 times, and it cost me, I think, $10 to get Zoltar's predictions of what is the field of cell death in the future. So yeah, $10, and that's a lot of money, right? Like a coffee. So, all right. I asked what is in the future of cell death research, and here's what he said. Zoltar says, in the future, we will not only count the cells that die, but also the cells that live. We all know this. Every time we write a paper or we do a project or we report our results, we say, oh, this worked, 60% cell death. But we don't say 40% cell life. That's our future, and we'll spend a little time talking about why. So, since 1970, there's a huge number of papers published on cell death. This is the percent of all publications. There are now 3 million publications a year, a total, altogether, and upwards of 3% of those include the words apoptosis, necroptosis, pyroptosis, or ferroptosis. There's a huge number of papers on cell death. But there are very, very, very few papers on cell survival, less than 100, less than 10, really. What happens when you treat cells and a lot of them die, but what happens to the cells that didn't die? So that's the project that we launched into a few years ago, the cells that live. Halimi Kalkavan really did the majority of this. She's a practicing medical oncologist who is doing her advanced work in research work in our lab, just finishing up, and Mark Chen, our computational biologist. So important for so many things we do. Now, when I said there's very few papers, the papers on the cells that survive are really interesting. Going back several years, it was noticed that if you treat a population of cancer cells, clonal population of cancer cells, with a chemotherapeutic drug, many cells die, but some cells don't. But they're all genetically identical, and they were all treated with the same drug. And what was noticed is that these cells take on new properties, the cells that live. And they're called persister cells. In this review, the deadly survivors. These persister cells, regardless of what chemotherapeutic was used, and really what cell line was tried, they have common features. And among those features is a condition of transient drug tolerance. So don't be confused. This is not selection for a mutant cell. These cells temporarily become drug tolerant and then become sensitive again. So for example, we engaged in this project using BH3 mimetic drugs. Now, I think you've heard about BH3 mimetic drugs. You know what these are. These are targeting the anti-apoptotic BCL2 proteins. They're exquisitely specific, and they kill cells in a back-to-back dependent manner. And we wanted to know not about the cells that die. A lot of great work has been done on that. How do these kill cells? But really, why don't they kill? What happens when not all the cells die? So, easy enough experiment. We treated with a combination of BH3 mimetics so that we would basically neutralize BCL-XL, BCL2, and MCL1 in the cells we were studying, and we used a lot of different cells. And most of the cells die. If you use enough concentration, all the cells die. But we used a concentration where some cells would live. And here's what happens. Those blue dots, those are the cells that grow. So that shows us living cells. So you treat the parental cells with the BH3 mimetic combination. The cells mostly die, but a few cells are living. And now you wait a day and treat them again with the same BH3 mimetics, but they don't die. They're now transiently resistant. But if we wait 6 days to see if what happens with the BH3 mimetics, now they're just like the parental cells. They die just the same. There's a transient state of drug tolerance. And in fact, here's just a graph of that on the left, but there's that transient tolerant state, but they're also relatively resistant to other chemotherapeutics. So for instance, using different cell lines, HT29, PC9, HT29, again, these are lung cancer and colon cancer, human colon cancer cells, but treating them either with general chemotherapeutics or targeted chemotherapeutics, those persister cells that survive the BH3 mimetic are somewhat more tolerant of other drugs that would induce apoptosis. But it's reported, and we've confirmed, that they also take on other changes. And among those changes is a sensitivity to cell death induction by ferroptosis. Now, again, I think you know something about ferroptosis. This is an iron-dependent cell death is driven by the Fenton reaction, where iron causes the peroxidation of lipids. And these lipids are detoxified by an enzyme called GPX4. If we use a drug called RSL3 against GPX4, it can engage ferroptosis if the cells are sensitized. And these persister cells are in fact sensitized to ferroptosis. When we look at the parental cells in the black lines, they're quite resistant to treatment with RSL3, or relatively resistant. But those surviving persister cells that resist other drugs are now more sensitive to RSL3. So there must be gene changes that are going on in these cells. And in fact, if we look at the cells that survive, the persister cells that survive treatment with the BH3 mimetics or with targeted or general chemotherapeutics, the emergent differences in gene expression look very, very similar. These are gene set enrichments of the various genes that change, some common differences that occur. And among those differences are a lot of genes that seem to control tissue colonization and invasiveness and metastasis. And in fact, when we look at the persister cells compared to the parental cells and inject them into mice, the persister cells are much better at colonizing and forming tumors in the mice. And you can see that here. This is now with HCT116 persister cells compared to the parental cells. And these are labeled with luciferase. So you can really see that the parental cells will form cancers when injected into these animals, but the persister cells are much more invasive. They're growing and they're more invasive, they're more metastatic. And I think you can see that pretty clearly here. We can look at this really in detail by two assays. One is an assay for intravasation, that is the movement of cancer cells from a tissue into the blood. Or we can look at real metastasis, where the tumors are placed in one lobe of the lung, and then we look for tumor cells in the other lobe of the lung. And in both cases, the persister cells are better at entering the blood and are better at metastasizing. So what a paradox. You treated the cells with a treatment to kill them, but they're now becoming more aggressive cancers. We're going to explore that a little bit. So what's going on? Of course, to really look at this, we have to understand the mitochondrial pathway of apoptosis. Again, I think you really know it, so I'm going to show it in real Brazilian style in the mitochondrial mambo. So anti-apoptotic proteins inhibit, and pro-apoptotic proteins, Bax and Bac, and sometimes Bach, promote the permeabilization of the mitochondrial outer membrane to release cytochrome C. And that activates APAF1, which in turn activates caspase 9, and caspase 9 cleaves and activates the executioner caspases 3 and 7, and we get apoptosis. Now, if you get rid of Bax, Bac, and Bach, of course, you can treat the cells, but they don't engage the mitochondrial pathway of apoptosis. So there's no cytochrome C release, but there's actually a drug called raptinol, which will permeabilize the mitochondria even of Bax, Bac, Bach triple knockout cells and promote cytochrome C release and caspase activation. It's described in the Cell Reports paper and confirmed in some other papers as well. So we're going to use that a little bit just to test some of our hypotheses. So we used to think that once you engage the mitochondrial pathway of apoptosis, that's it. The cells are going to die. In fact, Gustavo published the first paper on this, showing, even before we really understood the mitochondrial pathway of apoptosis, that if you engage apoptosis and then block caspases, the cells still die. We said it's a point of no return. But then a few years later, really more than 10 years later, Anna Kalel and John Ritchie, together with Stephen Tate, published that, in fact, cells that have released cytochrome C can still survive. And subsequently, Stephen showed that they can survive if some of the mitochondria are still intact. So if not all the mitochondria release cytochrome C, those cells can potentially survive. And that's going to be really important. So is the mitochondrial pathway of apoptosis important here? Well, one way we test that is we treat with the BH3 mimetic drugs. We see this increased metastasis and colonization by the persister cells. But if we get rid of Bax and Bach by siRNA, or we get rid of Bax, Bach, and Bach by knocking them out, treating with the drugs does not produce this phenotype. So that's a hint. That's also shown down here where we use these are the controls. These are the knockouts, which really don't show any of that increased metastasis. But if we take the triple knockouts and we treat them with raptinol to induce mitochondrial permeabilization, the cells that survive, once again show that increased metastatic potential in vivo. And it turns out that increased sensitivity to the inhibition of GPX4, that increased sensitivity to ferroptosis by treatment with the GPX4 inhibitor RSL3 is seen in persister cells, but not seen in cells that lack Bax, Bach, and Bach, but are treated with the same drug. So is it really the cells that have started to engage the mitochondrial pathway of apoptosis? To address that, we used a strategy that Stephen Tate suggested, but Halame managed to implement, that works like this. She attached a M-cherry to the intermembrane protein facing the intermembrane space in mitochondria, using a targeting sequence from AIF. But coupled to that M-cherry is a split fluorescent protein that's superfolded, but has no fluorescence. It's missing one piece of the fluorescent protein. And then she transduces that missing piece into the cytosol. If the mitochondrial outer membrane becomes permeable to release cytochrome C, this little piece, which is about the same size as cytochrome C, will now go into the mitochondria, bind to that superfolded GFP, and now make it fluorescent, and it'll turn green. Crazy idea, but it actually worked. So here you can see cells that are treated with BH3 mimetics, but then a full day later, when any dead cell is gone, we can see cells that have permeabilized their mitochondrial outer membranes and are fluorescing green because of this split fluorescent protein. This is quantified here by FACS, that with the BH3 mimetics, we see that shift in green fluorescence, which we don't see if we don't have Bax and Bach. But even in these Bax-Bach double knockdown cells, treating with raptinol, which is Bax-Bach independent permeabilization, again, we see that green fluorescence in both cases. So it really seems to work. But this lets us sort the cells that have permeabilized their mitochondrial membrane. And when Halame did that, sorting the cells that have the GFP fluorescence versus the cells that don't or have very low GFP fluorescence, the ones with the higher GFP fluorescence, the ones that have permeabilized some of their mitochondria, are also more metastatic in vivo. So that's kind of a mind blower. How in the world is that working? Okay. By the way, we also observe that there are changes in gene expression that depend on Bax and Bach. And these include features that are kind of a hint. They include, for instance, epithelial mesenchymal transition, which is also known to be associated with metastasis and invasiveness. And also a decrease in the component in genes that are involved in glutathione metabolism. These, which may account for why they're more sensitive to the inhibition of GPX4. We can do single cell RNA-seq on these cells at different times. And what we see is a pseudotime trajectory that's also evidenced in real time from the one-day persister phenotype moving through to the what looks like the parental phenotype over a period of 7 days. They revert. You can see that here, that the day 1 persister cells have a very different transcriptome from the day 7, which is approaching that of the parental cells. And when we interrogate those changes, one of the hallmarks that we pull out is changes in what's called the integrated stress response, which is high in the 1-day persister cells and gradually comes back to look like the parental cells where it's low. The integrated stress response works like this. Lots of different stressors, ER stress, amino acid deprivation, viral infection, or deprivation of heme, engage kinases, and there are only 4 of these, the 4 that are shown here, that phosphorylate a translation factor called EIF2B. I'll show you in a minute how this works, but the phosphorylation of EIF2B, well, it's actually EIF2 alpha, sorry. The phosphorylation of EIF2 alpha stops general translation, but promotes the translation of certain proteins. Among those proteins is a transcription factor called ATF4. So these kinases become activated, they phosphorylate EIF2 alpha, the cell shuts down most of its protein synthesis machinery, but now expresses the transcription factor ATF4. That's the integrated stress response. So we looked. Phosphorylation of EIF2 alpha is a clear indication that the integrated stress response has been engaged. And when we treat cells with BH3 mimetics, very quickly those cells phosphorylate EIF2 alpha and translate ATF4. When we look at the ATF4 positive cells, we can see that BH3 mimetic treatment induces that expression of ATF4. Raptinol treatment also does. Remember, this permeabilizes mitochondria in the absence of Bax-and-Back, or presence or absence of Bax-and-Back. If we take triple knockout cells that don't have Bax-and-Back, the BH3 mimetics don't induce the expression of ATF4, but raptinol still does. 2B-ACT is a inhibitor of the integrated stress response, which clearly is blocking the expression of ATF4 as well. So here's where it gets crazy. Hallamade just made cytosolic extracts. We've known for years, since the late 1980s, that if you make a cytosolic extract from cells and just add cytochrome C, that activates APAF1 and induces caspase-9 activation, which we can see as the cleavage of caspase-9. And that's been known for a long time, since really the pioneering work of Zhaneng Wang. But addition of cytochrome C also induces the phosphorylation of EIF2 alpha in these cytosolic extracts. We'll see this in a moment. I'm getting ahead of myself, but if we knock out, so we make cytosolic extracts, we add cytochrome C, or we add poly-IC, which activates one of the integrated stress response kinases, PKR. Both cytochrome C and poly-IC induce the phosphorylation of EIF2 alpha in these extracts. But if we remove one of the other kinases called HRI, which we'll come back to, we lose that phosphorylation of EIF2 alpha, which can still be induced if we activate this other kinase PKR. Okay. Oh, I guess I took out the slide. We had identified in other ways that it was HRI that was the kinase that was engaged. So again, we can knock out HRI. When we do that, we now see that the addition of cytochrome C in the parental cells induces phosphorylation of EIF2 alpha, which is completely gone in the absence of HRI. And that's just some quantification of that. But we also made recombinant HRI, the kinase, and we made recombinant EIF2 alpha, mixed them together, and added cytochrome C. And cytochrome C, so one thing you need to know is that HRI is a constitutively active enzyme, but cytochrome C increases its activity. But HRI normally is inhibited by heme in cells. So when heme is present, HRI is turned off. But when cytochrome C is present, so when there's no cytochrome C, HRI phosphorylates AIF2 alpha. If we add heme in the form of hemin, we suppress that activity of the kinase. But in the presence of cytochrome C, we don't. So, is ATF4 important? Is the integrated stress response important? If we take away ATF4 and induce persister cells, we still have cells that survive, but they no longer show that drug tolerance when treated with chemotherapeutics. And they no longer show that increased susceptibility to GPX4 inhibition. And they no longer show that increased metastasis in vivo that we can produce when there's no ATF4 or no HRI, which would activate ATF4 translation. We don't see that increased metastatic potential. We can define a gene set that is BAX/BAC and ATF4 dependent. And if we interrogate a database of single-cell RNA sequencing from patients with lung cancer, we can see that our signature is elevated in the minimal residual disease patients. After treatment, those patients that have very few cancer cells are showing that signature, which goes away or is not there in progressive disease. And that's both for the genes that we see that are upregulated and for the genes that we see that are downregulated in these data sets. Our signature of persistence is present in patients that have been treated with therapeutics. And in addition, we also see the downregulation of that glutathione metabolism. That might be a hint that these cells are actually sensitized for ferroptosis. And nicely, we see an upregulation of the HRI reactome, the HRI response, genes that respond to the HRI kinase, which is being activated by cytochrome C. So altogether, the persister phenotype is induced by BH3 mimetics, and persister cells are more invasive and metastatic than parental cells. The PS phenotype seems to be dependent on mitochondrial outer membrane permeabilization in the mitochondrial pathway and cytochrome C. Evasion of apoptosis after MOMP allows cytochrome C to induce an integrated stress response via HRI, and HRI and the resulting ATF4 are required for the persister phenotype. So when it comes to apoptosis, what doesn't kill you makes you stronger. When apoptosis fails, cytochrome C is released, but it can activate HRI, translation of ATF4, and produce this persister phenotype. Now, there's another aspect here, which Stephen Tate has worked on quite a bit, based on studies going back really to 2010, that cells that activate caspases but don't die cleave caspase-activated DNAs, and this produces DNA damage in these persister cells, leading to mutation. We're studying that as well, but I won't go into that now because it's time. These are the people who did the work, but I mentioned them along the way, and it's time to ask Zoltar another question. So Zoltar says, in the future, cell death pathways will engage other signaling pathways. I'm actually going to ask this a little bit in the context, and we don't know all the answers here yet. This is a fascinating question. Why don't apoptosis-resistant caspase-8 mutant mice, which I'll introduce to you in a bit, not develop LPR syndrome? And I'll tell you what all of that is. So I'm sure you know, because you're in a cell death course, that Fas ligand or CD95 ligand engages its receptor Fas, which causes the very rapid formation of a complex between FAD and caspase 8. And that leads to caspase 8 activation and apoptosis. If you don't have Fas ligand, or you don't have Fas, either as a human or in a mouse, this causes a really dramatic disease. In humans, it's called ALPS, acute lymphocytic proliferative syndrome, or lymphoid proliferative syndrome in mice, lymphoproliferation syndrome in mice. It's caused by defects in the Fas CD95 signaling pathway. They have, these children and young mice have enormous splenomegaly and lymphadenopathy, big lymph nodes. I'll show you those in a moment. And this is because of a weird population of T cells that have both T cell receptors and a B cell marker, B220, both in mice and humans. These animals become autoimmune. They also have an increased risk of cancer and a significantly reduced lifespan. The children usually require bone marrow transplantations, but there are some drug treatments that can be used for these kids. Now, 12 years ago, Andrew Oberst in our lab showed that we knew for quite a long time that you can't make a caspase 8 knockout mouse. But if you knock out caspase 8 and knock out RIP kinase 3 or MLKL, blocking necroptosis, now you get a perfectly good mouse, and they're born at Mendelian frequencies. But after a few weeks, they develop exactly this syndrome of the LPR syndrome, or acute lymphoproliferative syndrome. Huge lymph nodes, huge spleen. Now, we know from work that was originally started by Guy Salveson and then beautiful work from Marion McFarland, that the way caspase 8 is activated is that FAD causes a conformational change in caspase 8 by binding to the first DED domain. That exposes the second DED domain, which can bind DED1 in a second caspase 8 molecule. And this will cause an oligomerization of caspase 8 and its activation. And that's now understood at a structural level. Marion had shown that there were residues in the DED2 of caspase 8 that were required for this oligomerization. But interestingly, mutations in this F and L, mutating F and L to Gs, while it prevents caspase 8 interaction, caspase 8 flip interactions can still occur to make the caspase 8 flip heterodimer. So if you have a cell where the caspase 8 is mutated at these two residues in the second DED domain, they can only form caspase 8 flip heterodimers, which Guy Salveson showed are catalytically active. Indeed, we could make cells like this. We could make cells where we only introduce the non-oligomerizable caspase 8 or a catalytically dead caspase 8. If we treat them with Fas ligand, with cyclohexamide or other conditions to induce death, we can see in the cells with wild-type caspase 8, they die perfectly well in response to Fas ligand. If you don't have active caspase 8, they don't die. But also, if the caspase 8 can't oligomerize, there's no cell death. But cells like this don't spontaneously die by necroptosis. So if you have a cell that doesn't have caspase 8, and you activate through poly-IC, for example, that will engage necroptosis. You don't have to add a caspase inhibitor because caspase 8 is not present. But in cells with the mutant of caspase 8 that can't oligomerize, that caspase 8 flip heterodimer seems to be blocking necroptosis induced by poly-IC. So we made these mice. We mutated caspase 8 in the mouse, and they were born at Mendelian frequencies. Mice are fine. They're remarkably resistant to ligation of FAS. So you probably know if you inject an agonistic anti-CD95 or anti-FAS antibody into a mouse, it rapidly kills them in 6 hours. Almost all the mice are dead. If these animals, though, don't have any caspase-8 because they're a caspase-8 RIPK3 double knockout, they all survive. But the animals that have this mutation in the DED2 in caspase-8, they're pretty resistant to FAS ligation. They have a defect in the FAS pathway. And yet, they don't get ALPS. So if we have the double knockout caspase-8 MOKL animals, they accumulate in their blood these CD3 positive, B220 positive cells and get big spleens. But if they're carrying that FGLG mutation on the MOKL knockout background, or if they don't have if they do have MOKL, it doesn't make any difference. These cells don't accumulate and they don't get splenomegaly or lymphadenopathy. So can it be that that complex of caspase-8 and FLIP is sufficient to prevent this LPR disease? It happens in people too. There's a problem though with finding out the answer to that. Caspase-8 and FLIP, encoded by CFLAR, it's unfortunate the names are quite different. The gene name is CFLAR, but we call the protein FLIP, are very close to each other in the genome, right next to each other. So the only way we could actually do this would be to take our mutant animals and we cross them to MOKL knockouts just to make sure, and then knocked out C-FLIP in these animals. So knocked out the CFLAR gene. It works, and the animals are born at Mendelian frequency, which is great. But now they get ALPS. So that caspase-8 mutant animal never gets ALPS. If you knock out FLIP, now they do. Huge lymph nodes, big spleen, and the accumulation of these TCR positive, B220 positive cells in the circulation. So yes, caspase-8 FLIP complex controls LPR disease. So are they exactly the same as double knockouts? So we can compare the caspase-8 FGLG MOKL knockout to caspase-8 MOKL double knockouts. In this case, we're looking at RIPK3 knockouts because they happen to be available. The accumulation of those T cell receptor positive B220 positive cells is really obvious in these animals, not present in animals that are carrying the one allele of FLIP or one allele of caspase-8. But there are differences. So for example, in the caspase-8 RIPK3 double knockouts, T cells seem to be fine. There's normal T cells are fine, the ones that don't carry B220, plenty of B cells, no big expansion of myeloid cells. But in these FGLG, MOKL, FLIP double knockouts, you see a big accumulation of myeloid cells and a dramatic loss of B cells. And we don't really understand it, but we're learning something about it. One thing that we might consider was it might represent what Seamus has called the fatazome. And I don't know if Seamus talked to you about that, but Seamus showed that caspase-8 FAD and RIPK1 form a complex, not for signaling cell death, but for activating NF-kappa B. But Bart found that the FGLG mutant caspase-8 doesn't induce this so-called fatazome. If you treat cells with CD95 ligand and look for the production of TNF, which is one of the outcomes of this fatazome, wild type cells do it, but cells with this mutation in FGLG do not, and caspase-8 knockouts do not. Recently, though, there's another caspase-8 target that we've become interested in called N4BP1. N4BP1 is a protein that inhibits signaling from toll-like receptors. When you engage a toll-like receptor that doesn't activate caspase 8, inflammatory cytokines are blocked by N4BP1. But if you have a TRIF-dependent TLR, like TLR3 or TLR4, the engagement of TRIF activates caspase 8 via RIPK1 and FAD. N4BP1 is cleaved and inflammatory cytokines are now produced. And that was work that was published by Vishva Dixit's lab in Nature in 2020. They also showed that engaging TNF receptor would also activate caspase 8 to cleave N4BP1 and allow the production of inflammatory cytokines, even if the TLR was TRIF independent. So we looked. In our mutant animals that are carrying the caspase 8 that can't oligomerize, activation with Fas ligand or with LPS nicely induces the cleavage of N4BP1 in cells. Myeloid cells are what express N4BP1. However, if we get rid of FLIP, the CFLAR gene, now Fas ligand engagement or LPS engagement fails to cleave this protein. Now, Bruce Beutler published recently that the way N4BP1 negatively regulates cytokine production is by inhibiting NEMO oligomerization in the NF-kappa B complex. However, when we look at activation of NF-kappa B by the phosphorylation of P65, we really don't see any effects in terms of whether or not caspase 8 is caspase 8 or caspase 8 flip are present. We can't really, even though we see the cleavage of N4BP1, we don't really see this regulation of phospho P65. But of course, NF-kappa B is not just P65. There are a lot of NF-kappa B dimers that are transcriptionally active. P65 is also called REL-A. There's also a REL-B, and there's a CREL. A couple of years ago, our friend Igor Brodsky published that in a very different setting, caspase 8 promotes CREL-dependent inflammatory cytokine expression, and that got us thinking. So we take our cells that from the animals that have mutant caspase 8 or wild type or have mutant caspase 8 or have mutant caspase 8 and lack FLIP, and look for the induction by toll-like receptor signaling of CREL nuclear localization. And as you can see, in the absence of the FLIP caspase 8 complex, there's no nuclear translocation of CREL in response to Fas ligand, or in response to LPS, or in response to the to another TLR agonist in this setting. So caspase 8 FLIP really is regulating, probably through a N4BP1, but we don't know that, the activation of CREL. And whether that's contributing to some of these phenotypes we're seeing, we don't know. But in keeping with that, if we treat cells with Fas ligand or LPS or our TLR1/2 agonist, PAM3, the absence of the caspase 8 FLIP heterodimer prevents the expression of TNF. So that, again, I mentioned the people who did that work, although I do want to highlight that Scott Brown has been our collaborator in making all of these interesting mutant mice. Okay, time for another one. Zoltar says, in the future, cell death pathways will connect to all aspects of cell physiology. Now, this is going to be a thought experiment. It's called the button experiment, originally described by Stefan Kaufmann, a theoretical developmental biologist. He said, imagine you've got a table full of buttons, buttons like on your shirt. Now, pick 2 buttons at random and tie a thread between them. And now randomly pick up any one button. And of course, you'll only get one button. Repeat and repeat and repeat. Eventually, you have a bunch of threads and you pick up one and 2 buttons come up. And then 3 buttons. And then on average, 4 or 5 buttons. And you keep tying threads and you keep picking up a button. And in a computer model, you find out that when you have about half a thread per button, or 1 thread every 2 buttons, not even tied together, just the number of threads, you start to get a phase change. You start to pick up more and more buttons with each round. When you get to about 1 thread per button, it's already at the top of that phase change. You're picking up most of the buttons. Now, what does that mean? It means that if we tie together 2 proteins because they interact or they affect each other, and if each protein is connected to, on average, 1 other protein, most of them are connected to each other. And of course, we know we have proteins that are connected to many other proteins. Everything is connected. But also, the buttons don't have to be proteins. They could be metabolites, lipids, nucleic acids, ions, microbes, viruses. Anything that interacts with another element in our signaling pathways connects us to all other physiological functions. And we're only starting to recognize that complexity. So as we're doing cell death experiments, consider looking at other aspects of cell physiology and how our cell death pathways may connect. Zoltar says, in the future, dead cells and their engulfment will change the local environment. And in fact, we know an emerging and exciting field, it's been around for a while, but it's really exploding now, is the field of efferocytosis and how eating dead cells affects the microenvironment. Zoltar says, in the future, we will drug cell death pathways for therapies. You heard from Domagoy, and of course, he's interested in the inhibition of RIP kinase 1. We can inhibit BCL2, BCLXL, and now MCL1, but we can inhibit CIAPs. But what other inhibitors will we find? And now there are reports of inhibitors of BACs or inhibitors of BAC. We have inhibitors of other aspects or activators of other aspects of the cell death pathways. And we will drug these for therapies. We already are. In the future, many new cell death pathways will have roles in biological processes. We talk about apoptosis, necroptosis, pyroptosis, and ferroptosis, but there are other mechanisms out there. In fact, many ways that cells can die. And we've only scratched the surface of what the importance of some of those pathways might be. A paper that was just published showed that if you take away BACs, BAC, BAC, caspase 8, and MLKL, you can still make a mouse. Mice can still survive through development. There's a lot we don't know. In the future, analysis of cell death pathways will inform precision medicine. Laboratories like that of Tony Latai are using assays for the engagement of cell death pathways to predict which BH3 mimetic might function in the treatment of a cancer, but also whether or not they will synergize with conventional or targeted therapeutics. In the future, cell death pathway proteins will have other functions. Really, do each of these proteins only do the one thing we've looked at? Or do they have what we like to call day jobs? Their night job is to function in cell death, but what's their day job? What else do these proteins do in these pathways? We have so much to learn. And I'm going to end with this one, because this is a favorite puzzle that I've been thinking about for years. In the future, we will explore how cell death pathways evolved. We don't really think about that very much. Let me tell you what I mean. Invertebrates, and we know this is true in mammals, birds, amphibians, and fish, there's the mitochondrial pathway of apoptosis. It is the permeabilization of the mitochondrial outer membrane by BCL2 family proteins and regulated by BCL2 family proteins that releases cytochrome C to activate APAF1 and activate caspases. We know it works the same in all vertebrates. But a few years ago, we wondered if this is only true in vertebrates. And this paper, really from 10 years ago, was the work of Cheryl Bender in the lab, who was a graduate student. What she did, and it was rather difficult, was she showed that exactly the same pathway of the mitochondrial pathway of apoptosis, regulated by BCL2 family genes, activated by the release of cytochrome C to activate kinases, was present in echinoderms and platyhelminths, sorry I spelled that wrong, worms, and maybe in Cnidaria. Those are corals and jellyfish. Platyhelminths are flatworms, like planaria. Echinoderms are starfish and sea urchins. It's present in these. But in insects, it works very differently. In insects, mitochondria don't seem to be involved in apoptosis. The apoptosome is constitutively active, but the caspase that's activated by APAF1 is held in check by IAPs, and apoptosis occurs when an inhibitor of the IAP is expressed in the cell. Still more or less the same pathway, it's just cut short a little bit. But in nematodes, it's completely different. In nematodes, BCL2 does not regulate the permeability of the mitochondrial outer membrane. There's no BCL2 effector to permeabilize the mitochondrial outer membrane. Instead, the BCL2 protein sequesters the APAF1 protein and prevents its activity. When it's displaced by a BH3 only protein, now that APAF-like protein forms an oligomer that activates the caspase. But that's kind of weird because in us, BCL2 doesn't bind to APAF1 and inhibit its function. BCL2 prevents the release of cytochrome C that activates APAF1. And this is real. The crystal structure of the BCL2 protein from nematodes really binds to the APAF1 protein of nematodes. But the BCL2 proteins in us do not bind or inhibit APAF1. How in the world could that have evolved so differently? When we look at the tree of multicellular animal evolution, we can see that the animals that have BCL2 regulated mitochondrial outer membrane permeabilization and cytochrome C activated caspases sit in different arms of the evolutionary tree. Flatworms and echinoderms. Echinoderms are now very close to vertebrates on this kind of a tree, which of course vertebrates also have this. But nematodes have a BCL2 regulated caspase activation. That's quite different. And insects, arthropods, have lost BCL2 regulation altogether and regulate apoptosis a little differently. Something interesting happened here in the evolutionary tree to change how BCL2 works and how caspases are activated in cell death. We have no idea what's happening in some of these other organisms like crustaceans, or in some of these organisms like mollusks. We just don't know. So it's a great question. I don't think it's something we can solve by looking at sequences. We can only understand this by exploring protein functions, or maybe by computationally folding the various BCL2 and APAF1 proteins and looking for their interactions in silico. We'll see. So finally, these are all important and they're all matters of life and death. You can think of many more, I'm sure, in your own research. But Zoltar says, in the future, we'll have classes in person. And hopefully that's what'll happen next time. Thanks a lot, you guys.
All right, man. What a way to close this graduate course. I mean, I told in the beginning, this guy is an inspiration. No, really, it's incredible. Every time
You are lucky and we are lucky.
Yeah, every time I hear from you, I get motivated because, you know, we've been through a series of events that just make us think, wow, let's just close the lab and do something else. But then, thankfully, there comes you to our rescue. Okay, guys, turn your video on. Let's chat a little bit with Doug.
Yes, he has only a few minutes.
Oh no, I have plenty. I have almost half an hour.
Okay.
Can I just start with something, Doug? When you start to presenting your persistent cells and then you talk about the integrated stress response, I actually thought that the answer would be autophagy. So is that autophagy playing any role on this? Is that connected well in this your big network?
Yes.
Or
we're just finishing those experiments, so I couldn't show you. One of the reviewers on this paper wanted to know, how are the cells tolerant of apoptosis? And we observe in these cells, the cells that survive have greatly reduced levels of BAX. And those reduced levels of BAX are responsible for this transient resistance. But it's not a change in the expression of BAX at the mRNA level. It's just the protein. Now, it's known that ATF4 can activate autophagy. So it's not a perfect experiment, but it's as far as we have right now, that if we block autophagy, now we restore BAX levels in these cells. And this is only blocking lysosomal degradation. We block BAX. So it could be that one of the first things that happens is ATF4 driven autophagy is depleting the levels of BAX and making those cells more tolerant of the drugs.
So this is a feedback mechanism that first you get the integrated stress response, and then autophagy. Autophagy is not happening at the same time. Let's say if you block autophagy, would you get less persistent cells?
That's the suggestion. We haven't done that, but yes, that's a prediction of this. Yes, and in fact, we could show, for example, that the cells that don't have ATF4 or don't have HRI don't lose their BAX level.
And how is the sensitization of these persistent cells to other pathways, such as necroptosis or pyroptosis?
Yeah, these cells, the tumor lines that we're using aren't sensitive to necroptosis because, as you probably know, tumor cells that are adapted to culture tend to lose RIPK3 or MLKL, and these have. So we haven't interrogated that. We really only looked at ferroptosis because of the literature that said that the cells are sensitized to ferroptosis, and we could reproduce that. Yeah.
Okay.
That's also, it's incredibly interesting because apparently, so all our system that prevents ferroptosis is something that has to be very ancient, because that's what we, the insult that we get, in contact very often. So now you get resistant to everything else but sensitivity to the fructose. So it looked like the nature is always compensating
It
somehow.
does. It looks like that. Now, a lot of people are really focusing on the ferroptosis to see if that would get rid of the persister cells, that if you combine therapy, there's a lot of focus there. But we're starting to think that maybe inhibition of autophagy or inhibition of the integrated stress response might be a better way to go. You probably know that the drugs that induce ferroptosis in vivo are not very good.
You mean for these resilient cancer cells that recover from that?
Right.
Yeah, no, it's.
That's,
So we are pretty close. We're getting close to the cure of cancer.
well, let's hope. you know, it's we're curing more cancers today than we ever have, but there's so many more to cure. And I'm not sure this will be true in all cancers, or this we don't know at this point. We sort of focus mostly on lung cancers in this story, but also some colon cancer as well. But these were just cell lines, and as you can imagine, the big challenge was, can we actually identify cells that have started to engage the mitochondrial pathway? The mitochondria are permeabilized, and that was that great probe system that let us do that. And then to see, how do those cells change if they don't die? And that's been our challenge. Yeah.
Yes.
That's very nice. What about the BCR-ABL? You haven't researched anything, yeah? Because that's also kind of a now is resurfacing, studies on the kinase resistance and
Oh,
the
absolutely.
stem cells. So that may apply with that.
Absolutely. Well, you're the one who showed that BCR-ABL inhibits apoptosis.
Just one of the person.
Yeah.
1 year ago.
Long time ago. I think David's got a question.
Yes,
All right.
David. Go ahead, David.
Much, Professor Green. Great lecture. So about the ER stress that you reported on these persistent cells, you showed data about the PERK branch, but I would like to know about the other branch that for the protein response can be activated, like the ED1 alpha phosphorylation or the ATF6 cleavage. These branches are also activated, or the PERK
No.
branch is the only one?
No, so if you saw, it wasn't PERK that was being activated. It was HRI, which is EIF2 alpha kinase 1. PERK is EIF2 alpha kinase 2, I think. So we don't see the activation of PERK. We don't see ER stress. We see a cytochrome C induced integrated stress response that's dependent on HRI. Now we can produce ER stress. We can produce ATF4 activation by ER stress. And we actually do see that also increases metastasis. So I think engaging any of the integrated stress response can have some of these features. But the phenotype in the persister cells, at least in the way we're doing the experiment, seems to depend on HRI.
Doug, I have another questions. Just waiting, until one of the other students raise hand. Is it because this response is transient does this account for the metastasis colonization in patients that receive treatments, things like that? Do you think that?
We think so. Of course, we haven't proven it. We think that's the case. A lot of other people have suggested. So Jeff Edelman, who made the original observation on persister cells, suggested that what this does is it gives an opportunity, because the cells survive longer and maybe are mutating, which Stephen would say is happening because of the CAD activation, that maybe now there's an opportunity to produce a real resistant mutant. So if you killed all the cells with the first treatment, you might not find the resistant mutant. They might arise after the treatment in some cases. They might not be there before the treatment. And this might be the case in some situations. So going back to imatinib and BCR-ABL, of course, if the first time you treated with imatinib, all the cells died, you've got no problem. If there was a mutant cell that's imatinib non-responsive, just by chance, if that cell was there, okay, it could relapse the tumor. But even if you don't have the mutation, if you didn't kill all the cells, now they have a window of opportunity to generate the mutants that will now ultimately be resistant to imatinib. Now, we don't know that's true, but it's something, it's a system we're very interested in exploring next, to start looking at this mutation that follows. Yeah. And we've reproduced Stephen's data that if we take the persister cells and look for increased mutation, it's there. And it's dependent on CAD. So.
Oh, it is dependent on CAD. It's like a circle, full circle.
It's like a full circle, all that old stuff. Yep. See, I told you, it's really what this is if you turn it on its head and you say, let's imagine that cells can survive the cell death pathway, what are the consequences? It's a whole new world of investigation.
Yeah, exactly. Really. Cool. That gives us a lot of things to think about it. Cool. Would you think so? So this, you're doing like apoptogenic stimuli, huh? What about the other pathways? So you think that you can block, the inflammasome in the mid before, and then you get some of these as well?
I don't know if it'll be the same changes. We showed a couple of years ago, Yinan Gang's paper in Cell 2017, so wow, 5 years ago. He showed that in necroptosis, cells can activate MLKL, but if you turn off RIPK3, or if you inhibit MLKL after MLKL is activated, the cells can still come back and survive. And they repair their membranes. They repair the plasma membrane in an escort dependent manner. So escort is a molecular machinery that can repair membranes. And he showed that that was what was doing it in necroptosis, that cells can activate necroptosis but not die. Yeah. Yeah.
That's pretty cool.
Yeah, and actually, Peter Broz and Sebastian Ruhl published in Science after our paper. They published that in pyroptosis, also you can activate pyroptosis, but if it's not complete, if the cell, even if it has gasdermin D, the escort machinery can antagonize the gasdermin D pore and repair those cells and keep them alive.
Yeah, that's another, field to look at, resistant cells to, whatever bacteria or pathogen, and see how they behave in the second round of, infection or things like that. So it looks
exactly.
Really
now,
cool.
Now
Yes.
you might be wondering, why in the world would the system be built like this? Why would you build the system so that if you have a cancer cell and it doesn't die, it gets worse? That, you know, how did that come up in evolution? And my answer to that, which is only hypothesis, don't know, but it makes sense to me if this is really about wound healing. So imagine it, wound healing, tissue
wound
damage.
healing, yeah.
Wound healing, yes.
So imagine you have a tissue and it gets damaged by some chemicals or environment or whatever. And now
Cells.
it's in a race because the cells of that tissue have to repair the tissue. But if they don't repair it fast enough, fibroblasts will proliferate. But of course, fibroblasts are not functional tissue. So, you know, that's fibrosis and that's a bad thing. So maybe you kill, you know, you have this environmental insult, a lot of cells die, but some cells at the edge of that don't quite die, but they become now much more aggressive and they invade and they metastasize and they fill in that tissue now and heal it. So maybe this is this response is a way to say, oh, I didn't die, but that it was probably killed my neighbor. I need to repair. Maybe.
Yeah, makes sense.
Fantastic, yes.
Yep.
Doug, jump into the last part of your talk for the evolution. I don't know, I don't remember actually how this caspase 1 evolved during this time. And also for the necroptosis machinery, I don't heard a lot about it.
Oh, no, both are incredibly interesting. So with caspase 1 and caspase 11, it's wildly different. So even, so you know that, of course, caspase 1 cleaves IL-1 beta and IL-18 and also cleaves gasdermin D. Caspase 11 or caspase 4 and 5 in us can cleave gasdermin D, but they don't process cytokines, right? But in dogs, there is no caspase 1, caspase 4, caspase 5. It's a caspase that looks like both of them, and it does both. It's activated by LPS, and it cleaves IL-1. But in cats, it's activated by, I can't remember. It's a similar thing in cats, but it has a different function. And this was work John Kagan published recently. It's
John
really
Kagan.
Kagan,
Yes,
yeah.
I remember this.
So it's
yes.
really crazy stuff. But here's the question. I mean, we love the idea a bacterial product directly activates inflammatory caspase. So caspase 11 or caspase 4, caspase 5, LPS directly activates it. Okay, cool, which is really fascinating, right? You would think that this would be, why just LPS? You would think, well, it's a good strategy. Why not have many different bacterial products activate different caspases? Well, here's a puzzle, no answer. Echinoderms have 42 caspases. So are some of these the sensors for different kinds of bacteria?
But are then inflammatory and apoptotic or anti-inflammatory?
No idea. It just comes out of sequence databases. They also have a huge number of inflammasomes. So
And
lots
huge
of
number
and
TLRs as well.
and lots of TLRs, yes.
Yeah.
So no idea how that's all wired. And, you know, we really haven't explored this in a lot of depth. You know, there's a few funny experiments now and then. People clone a gene out of some organism, but now it's pretty easy, right? Once you have the sequence, we can make the protein, we can put it in a cell and see what it does, or we can even make the protein and see how it functions. But there's really no work on this, very little.
Very.
Doug, I have another question for you that I know that you're not working directly on inflammasomes, but I'd like to get your perspective on this NLRP3 damn thing that are activated by so many signals and nobody figured this out. So what do you think?
No, if you notice, actually, there's a paper Nav Chandel just published where he said it's not mitochondrial ROS. He said it's not ROS. And some people say maybe it's mitochondria. Somehow it's something to do with mitochondria, but we really don't understand it. Michael Karen says mitochondrial DNA activates the NLRP3 inflammasome. But I have big issues with that.
Maybe mitochondria is a site to initiate the assembly of NLRP3 because they has this carbonylipin and
well, you know, there's really three big camps. Well, two big camps and one little camp. One camp says it's mitochondria somehow activate NLRP3. Some of them say ROS, some say mitochondrial DNA, some other thing, like Karina said. There's also the camp that says, no, it's all lysosomes. So if you perturb lysosome function, you activate NLRP3. And that's what
many of them don't believe that. I believe, but many of people don't believe.
I know, exactly. Yeah. And then there's a few people, including Vishva Dixit, who think, no, we've missed it. There's a completely different sensor that, like the NAPES, that works with NLRP3. I don't know.
Oh.
I don't have an answer.
Gabriel Nunes believes only in the potassium efflux.
Right. Yes, you're absolutely right. That's the other. The other
many signals actually do this. All these signals from mitochondria and the lysosomes and the reticulum actually do this potassium efflux.
Absolutely. And
some of them believe that. I don't believe.
But we should probably, I agree, but we should probably keep that. What is that really telling us? So when you reduce potassium levels, NLRP3 spontaneously activates. What that means is that somehow NLRP3 is held in an inactive conformation, but it can spontaneously achieve the activated conformation. And so that's probably telling us that there may be many conditions in the presence of potassium that can push the conformation through its energetic barrier to the activated form. So it may be correct that many things can push it this way. Possibly.
Possibly, yes, of course.
Yeah, and bizarrely, we are seeing roles for NLRP3 in so many settings. And it's just mind-blowing to us.
Many what? So
situations.
Serious. Okay.
So I'll just tell you one experiment that makes me crazy, but it's amazing. You may know a couple of years ago, we published papers showing that if you perturb the autophagy machinery, but it's not autophagy, it's actually our OT3-associated endocytosis, that this can promote Alzheimer's disease-like symptoms in mice. So we see all of the hallmarks of Alzheimer's disease when we perturb these pathways. So we take animals that are Alzheimer's mice that we've made, and if you measure the short-term memory of these animals, they have none. They don't remember anything. So we do a lot of different behavioral tests, and they're extremely deficient. Y-maze and novel object recognition and sucrose preference tests. And now we treat the mice. So we test them and they're like brain dead. Their memory is garbage. Now we treat the mice for 8 weeks with NLRP3 inhibitors. And now they're just like the normal mice. They're almost completely better. They get their memory back. This is crazy, right? It's crazy.
Yes, it's crazy.
But it's related to cell death or related to?
Well, I'll tell you a secret. When we cross our Alzheimer's mice to MLKL knockout, they don't get disease.
So that is
I don't know.
rather this, yes.
But do you see less cell death, in fact, or its function?
Yes. So we see massive cell death in the neurons of these animals. Okay. But when we treat with the NLRP3 inhibitor, it goes away. I mean, of course, the dead cells are gone, but we don't see any more cell death. Yeah.
Right. So I have some
cell death. We lose phospho-tau. So the tau protein is in neurons, is in axons, and it's phosphorylated in Alzheimer's disease. It's very strongly phosphorylated in our models, but if we treat with the inflammasome inhibitor, it goes away. Really dramatic, really, really dramatic.
Yes, Dougie Gollenbach demonstrated the role of NLRP3 only in microglia, not in other cells, to induce Alzheimer's-like symptoms. And it was related to the neuroinflammation, not at some point that I followed, but I don't know now. But at that point, it was related to the neuroinflammation and not to pyroptosis of the cells. So
I agree. I mean, that we also see it's all in the microglia. Yeah.
Yes. So there is a note here from our student Gustavo Martins. I don't really have a question to Professor Doug, but I.
Você quer falar, Gustavo? You want to?
Of course. I have just written in the chat, Professor, and I just wanted to make it official and say how inspiring your lecture has been, and that science is rewarding, of course, all by itself, but it is very rejoicing when you look into the future and you see how it may impact into science, to advance knowledge, to improve therapies for patients. And I think your lecture has passed some of that enthusiasm to all of us students here, and I would like also to thank Professor Gustavo and Professor Karina for joining this group of excellent professors and organizing this amazing course. It has been of great learning for everyone. I am sure of that. Thank you, Professor Doug, again. And
Gustavo, I really appreciate it. I appreciate that so much. And, you know, really this week, you guys have had some of the best people in the world speaking about cell death. These are really the best. And I'm glad it was inspiring, because it inspires me too.
All right.
Thanks, Gustav.
Anybody?
And I think I do have to go because I have to run my
yes
program.
So,
Doug, super thanks again. Thanks a million.
Thanks.
If the students want to stay a little bit more, we can just chat for 5 minutes. And thanks a lot, Doug. Super.
Take care. It's so great to see you. So fantastic to see you.
So great.
Thanks, you guys.
It's great.
Thanks.
Thank you.
Take care,
Thank you so much.
It was great.